All right, so Wonder Kids, let's get started today. We have a lot more fun activities, and we have our very own speaker for today. So we learned that hypothesis is a guess made up of all the backup information you know before doing a science experiment. So do you guys remember the other steps of the scientific method? So for the scientific method, we learned we need an observation, we need a question, we need a hypothesis, and we need also an experiment. After we do the experiment, we do a conclusion, and then we do the results. So do you guys remember what a microscope is? What do you use a microscope for?
Luca?
You use a microscope for like looking at very small things.
Yeah, really, really small things, right? And I think the Kai also has an answer. Go ahead, the Kai.
Oh, I guess he doesn't. Oh, go ahead, Dekai.
Go ahead, the Kai. No?
Oh, no, he doesn't. That's okay.
It's all right, the Kai. I know you got the answer right here. Yeah, so like Lucas said, a microscope is used to look at very, very tiny things like bacteria, which are germs, like our blood. So you guys remember on Wednesday, we looked at a strawberry underneath a microscope and how it looks so different from what you think it'll look like, right? And we looked at water bears, which are these really, really tiny animals.
All right, Wonder Kids. So for our experiment, we will be doing microscope lens, and we're going to see how these work. So what you'll need, go into your week 2 microscopy bags. Does everyone have their bags? Awesome. So look and see if you have the 3 materials that you need: a magnifying glass, Fruit Loops, and a fingerprint slide. So that's like a tiny clear glass rectangle.
So check it, like open it now.
Yeah. And then you're going to use that to see what the Fruit Loops look like with the magnifying glass. Hi, Rosalind. Hey, you're just in time for our experiment.
Yeah, you guys can make sure to draw your observations too in part B. Do you guys have any predictions on what you think it's gonna look like? Yeah, awesome. Yeah, so a magnifying glass makes things look a lot bigger. And a microscope is kind of like the same thing too. It uses a lot of these lenses, like the magnifying glass. It uses magnitude to make something that is pretty small into something a lot bigger. Look at, you want to share what you see?
I see it looks like if you use a magnifying glass over it, like at first you might not really see anything, but if you put a magnifying glass over it, you kind of see like little powdery stuff.
Powdery stuff, yeah, like powdered sugar, right? I would have never imagined it had like a lot of powdered sugar on it until I looked at it under a magnifying glass. A lot of scientists need magnifying glass to be able to look closer. Yeah, those. I want you to tell me what you see underneath this magnifying glass.
These Fruit Loops are big.
They are a little bit big. Do you think the magnifying glass will make it bigger or smaller?
It's gonna be bigger.
Yeah, right? Because of magnitude makes little things look a lot bigger. And we're going to learn more about that too.
I'm gonna eat it.
You can go ahead and eat it too. You guys can eat all the leftover Fruit Loops. All right, guys. So the next thing is going to be the fingerprint. Miss Mia, what are we going to do with the fingerprint?
All right, boys and girls. So you want to take your fingerprint slide, take one finger and press on your slide. And then after you do that, it's going to leave a fingerprint for you to check with your magnifying glass. And we're going to see what are the patterns that your fingers have. So go ahead and put one finger and press down on your fingerprint side. All right, so you're gonna take one finger. You can use either your thumb or maybe your index finger or your pointy finger. And then you want to take your fingerprint slide and just press down. Press your finger on it. Use your magnifying glass to see what it looks like up close. Who wants to share? Who wants to show us their fingerprint? Rosalind, do you want to share what your fingerprint looks like?
Yeah, my fingerprint looks like a thumb.
Okay, awesome. And then did you see what it looked like under the magnifying glass?
Yeah, it
What's
looks so big.
It looks a lot bigger, right? Can you see the
Yeah.
detail of your finger and the fingerprint and the different lines? Who else wants to share?
I noticed that when I put it up close, it looks like my hand is bigger.
Yeah, your hand's
my
bigger, right?
finger is bigger than it.
Yeah. Well, that's how magnifying glass works. You can see things more up close. All right, last one, Luca, go ahead.
I think it looks like your fingerprint looks like it's like really like swirly, but if you zoom up, it's actually swirly in two different directions.
Wow, that's a really good observation. So we can see exactly the direction of the little lines of our fingers, right?
So everybody give a big round of applause for our Wonderkid speaker, Mr. Sam.
Hi, Wonderkids. How's it going? Okay, so I'll be talking to you guys today about microscopes and a couple of fun things, give you a little bit of background on how I got into science, because I know many of you are interested in a lot of different things in science. And I want to say that it's very normal for those things to also change as you grow up. When I first started, I really liked dinosaurs, and especially I liked whales. So what got me into science when I was a kid, I started volunteering at a science center, and I really, really, really wanted to be a marine biologist. And then when I grew up, I went and did some research in marine biology. I already enjoyed scuba diving, so you can see me with some really big fish over here. And I still enjoy scuba diving, but I found as I did some research with marine biology, I didn't like it as much. And that's, a big and important thing for you guys to do, is always try things. And that way you will figure out if you do like or don't like something, because sometimes you might think you really enjoy something, and when you actually experience it, you don't. And similarly, sometimes you might think that something is not so fun or it's boring, but when you actually engage with it, you find that it's a lot of fun, maybe something that you really, really enjoy doing. And so, as your instructors are just saying, we have this DNA to RNA to protein chain of events. And I'll show you guys some cool microscope images that we've taken in the lab. All of them relate to the fly eye. So as your instructors were just saying, when you have your DNA, you can think of it as a hard copy, like a book, right? All the instructions that make you are in this book. And the mRNA will write down the instructions for whatever it needs to make, like your hair or your nails, so that nothing ever happens to the book. Things can happen to the actual notebook, the instructions that it takes, but nothing ever happens to the actual book, so that everything stays the way it should be. And then finally, those instructions can be used to make something, right? So they'll make a protein, but in this case, you can think of it like, any tool, a nut, a bolt, any of these types of things. Now, when we're imaging these, we use what's called a confocal microscope. It's the very same type of technology that you guys have. It's got some fancier lasers instead of using regular light, and it's very, very big. So you can see, for example, compared to a computer, that this takes up, half of a room. And part of that is because we need to see things that are very, very, very small compared to what you guys are seeing. And so here you can see, for example, let me pause it when it comes time. So here you can see, for example, this is the adult eye, and all of these rows, right? All these little eye units are all very nicely spaced. This has to happen when it's in the caterpillar form before it becomes an actual adult. So there's the DNA that has copies of it made into mRNA, and this is the mRNA essentially giving instructions so that all of these little teeny tiny cells that will eventually become the fly eye are all organizing themselves now, so that when it goes into the cocoon, all of them can actually make this beautiful structure. And we can do a lot of cool things when we image things like this, so we can see all these individual little bumps, and we can study how well spaced is this eye. And similarly, we can look at many different mRNAs all at the same time, which is very fun, very exciting, and each of them does something slightly different. So you can see, for example, this is one of the mRNAs. It's called atonal, and do you see how it has very nice spacing over here between all these little red clusters? Because this is the main one that decides where each of these gets placed. Some of these other ones, like Delta, like Harry, these are additional instructions. Once all of these guys have decided where they're going to be, these ones decide some of the cells and the other things that need to occur around that entire eye.
Oh, for example, the last Wonderkids group asked a very interesting question. So you guys know, like, the fruit flies, they love to fly around, right? They fly everywhere. You can't stop them. So somebody asked, like, oh, how do you stop a fruit fly from just moving around? Right? What do you do to stop the fruit flies so you can be able to look at their eyes?
Yeah, so when we need to make sure that they're not flying around, we usually have them in a jar. We can add a little bit of gas so they sleep for a little bit, and that way we can put them on a microscope slide, the same type of glass slide that you guys were looking at earlier, those little glass rectangles. And that way we can look at the fly and not have it move around. Other times we might need to perform a little fly surgery and actually look at the brain or the eyes or some of the internal parts.
Allison, you look a little shocked with the fly surgery. Go ahead, Allison.
So the surgery, you do it under a microscope?
You do, yes, and you use really small tweezers. I have a pair that I can grab in a second, but you use very, very small, fine tweezers, and you can actually very carefully, remove the actual brain to study it. So I am a brain surgeon technically, but I'm just a fly brain surgeon. Yes, Alvin.
About like how small are the organs, or like the brain, like how
The
small are
forceps or the brains? The forceps, yeah. I'll go grab them. Give me 1 second. Let me go find them.
Imagine the technique. You have to look over in the microscope, so tiny to be able to take out the little brain.
So you can see these are like your regular size tweezers that you use for things. When we're dissecting them, we'll use something like this. Do you see how much finer and much smaller? This thing almost comes down to like a needle. It might be hard for you to see. I'll see if I can get the reflection. But oh yeah, right there. Do you see how it shines at the way tip? So the way tip has a mirror on it, and part of that helps in making everything not sticky, but it comes to a really, really, really small, fine point. And usually you'll sit under a microscope with 2 of these and just very carefully move whatever tissues you need to do. And this is the same type of surgery that they need to do on any animal for like a vet or doctors, right? So they have to do all this very carefully with these very small tools, but they have to use the microscope so that you can see exactly where you are, and you have to have very steady hands.
Do you work with any other animals besides flies?
I've done work with a couple of different animals. I've worked with starfish and mussels. So when I first started studying here in California, I would go to Malibu and we would collect some of the sea stars, and they have, the mussels that are always on the rocks. And the reason that scientists study this system is because the sea stars will eat up the mussels. And when the mussels are removed, barnacles and crabs and many other animals can live on that rock. But if things happen to the sea stars, which climate change and some other things are now starting to harm the sea stars, because the sea stars are the main animal that eats things and allows space on the rock, so a lot of different things can exist. When the sea stars are removed or harmed or start dying out, then the mussels just take over everything. So then you only get mussels. You don't have crabs. You don't have barnacles. You don't have a lot of other diversity in that actual system. And that becomes a big problem because you need a healthy ecosystem where things are being recycled, and something eats something, and something eats that, and you have this whole full circle happening instead of one species or one thing kind of taking over.